Exercise hormone halts Parkinson's disease symptoms in mouse study. Researchers from Johns Hopkins Medicine and the Dana Farber Cancer Institute in Boston have shown that a hormone secreted into the blood during endurance, or aerobic, exercise reduces levels of a protein linked to Parkinson's disease and halts movement problems in mice. Parkinson's disease, a neurologic condition that causes people to lose control over their muscles and movements, affects about 1 million people in the U.S. if confirmed in additional laboratory research and clinical trials. The researchers' study in mice engineered to have Parkinson's disease symptoms could pave the way for a Parkinson's disease therapy based on the hormone irisin. Results of the researchers' tests appeared August 31 in Proceedings of the National Academy of Sciences. Johns Hopkins Medicine's Ted Dawson, MD, PhD, and Dana Farber's Bruce Spiegelman, PhD, worked together to look into the link between the exercise molecule irisin and Parkinson's disease. For unknown reasons, endurance exercise has long been found to alleviate symptoms of Parkinson's disease. Dawson, whose research focuses on neurodegenerative diseases, including Parkinson's disease, said one of the first clues to the link between exercise, Parkinson's disease and irisin came from Spiegelman, whose first paper about irisin was published in 2012 in Nature and subsequently in other scientific journals, showing that a protein called an irisin peptide is released into the blood and increases with endurance exercise. In the past decade, other laboratories have found that exercise elevates levels of irisin, and there is interest in looking into the connection between irisin and Alzheimer's disease as well as Parkinson's disease. To test the effects of irisin on Parkinson's disease, Dawson and Spiegelman's teams began with a research model used by Dawson in which mouse brain cells are engineered to spread small, spindly fibers of alpha-synuclein, a protein that regulates moods and movements related to the brain neurotransmitter dopamine. When alpha-synuclein proteins clump, those clusters kill dopamine-producing brain cells, a key trigger of Parkinson's disease. Fibrous clumps of alpha-synuclein are very similar, says Dawson, to what is found in the brains of people with Parkinson's disease. In the laboratory model, the researchers found that irisin prevented the accumulation of alpha-synuclein clumps and its associated brain cell death. Next, the research teams tested irisin's effects on mice engineered to have Parkinson's-like symptoms. They injected alpha-synuclein into an area of the mouse brain, called the striatum, where dopamine-producing neurons extend. 2 weeks later, the researchers injected a viral vector, which increased blood levels of irisin, which can cross the blood-brain barrier, into the mice. 6 months later, mice that received irisin had no muscle movement deficits, while those injected with a placebo showed deficits in grip strength and their ability to descend a pole. Additional studies of brain cells among the mice given irisin showed that the research team demonstrated that irisin also speeds up the transport and degradation of alpha-synuclein via fluid-filled sacs called lysosomes in brain cells. If irisin's utility pans out, we could envision it being developed into a gene or recombinant protein therapy, says Dawson, referring to the widening field of drug development aimed at using cellular genetics to treat disease. Dawson is the Leonard and Maudlin Abramson Professor in Neurodegenerative Diseases, Professor of Neurology and Director of the Johns Hopkins Institute for Cell Engineering. Given that irisin is a naturally produced peptide hormone and seems to have evolved to cross the blood-brain barrier, we think it is worth continuing to evaluate irisin as a potential therapy for Parkinson's and other forms of neurodegeneration, adds Spiegelman. Dawson and Spiegelman have filed for patents on the use of irisin in Parkinson's disease. Spiegelman has created a biotechnology company, Avum Therapeutics Inc., based in Boston, to develop irisin into treatments for neurodegenerative disease. Other scientists who contributed to the research include Tae In Kam, Hyejin Park, Shih-Ching Cho, Yu Ri Choi, Devanik Biswas, Justin Wong, Yu Shin, Alexis Loader, Senthilkumar Karapagounder and Valina Dawson at Johns Hopkins, and Jonathan Van Vranken, Melanie Mittenbuehler, Hyonu Kim, Mu A and Christiane Ran at Harvard Medical School. The research was funded by the JPB Foundation, the Maryland Stem Cell Research Fund, the Mark Foundation for Cancer Research, the Damon Runyon Cancer Research Foundation and Deutsche Forschungsgemeinschaft. Please consider subscribing to this channel.